Welcome to one of our gastro slash endocrine lectures where I talk about insulin, glucagon, and diabetes control. This is first a really excellent pancreas function Picmonic. Picmonic is a great way to learn difficult, challenging topics using pictures, and it creates a story like a mnemonic. So really helpful there. When we look at insulin synthesis, there's two amino acid chains that are connected by disulfide linkages. They are synthesized by beta cells in the pancreas. It's first created as pre-proinsulin. That's from translation from an RNA. Then pre-proinsulin is cleaved into proinsulin in the endoplasmic reticulum. Proinsulin has three peptide chains, A, B, and C. And in the Golgi apparatus, that's when proinsulin is cleaved into insulin, which has A and B chains, or alpha and beta chains, linked by a disulfide bond, as well as C chain peptide or a connecting peptide. Insulin is then secreted into the blood through secretory granules and it's unbound circulation in the blood. It has a half-life of really short, about 6 minutes, and it's broken down by the enzyme insulinase in the liver. C-peptide gets cleaved off of insulin and it binds to membrane G proteins to activate different enzyme systems. It can activate sodium potassium adenosine triphosphatase. It can activate endothelial nitric oxide synthase, and we can measure it. This can be helpful in type 1 diabetes to determine how much natural insulin are we producing. Within seconds after insulin binds to a membrane receptor, about 80% of the cells in the body will increase their uptake of glucose. Muscle and adipose cells excessively. Not neurons in the brain necessarily. They are not, well, not at all. Neurons are not triggered by insulin. They just get sugar as a first pass regardless. When glucose is transported into a cell, that's going to increase our energy production typically. The cell membrane also becomes more permeable to amino acids, to potassium, and to phosphorus. And our enzyme levels intracellularly are upregulated or adjusted for about 10 to 15 minutes after that glucose is moved inside. This also will then have a downstream effect of changing rates of transcription and translation. That's my little insulin brief video for you, a little taster to get started as we transition into more gastrointestinal content and endocrine content.